peroxide is a compound containing an oxygen-oxygen single bond or the peroxide anion, O2-2. The OO group is called the peroxide group or peroxo group. In contrast to oxide ions, the oxygen atoms in the peroxide ion have an oxidation state of A1. The simplest stable peroxide is hydrogen peroxide. Superoxides, dioxygenyls, ozones and ozonides are considered separately. Peroxide compounds can be roughly classified into organic and inorganic. Whereas the inorganic peroxides have an ionic, salt-like character, the organic peroxides are dominated by the covalent bonds. The oxygen-oxygen chemical bond of peroxide is unstable and easily split into reactive radicals via homolytic cleavage. For this reason, peroxides are found in nature only in small quantities, in water, atmosphere, plants, and animals. Peroxide ion formation has recently been highlighted as one of the main mechanisms by which oxides accommodate excess oxygen in ionic crystals and may have a large impact on a range of industrial applications including solid oxide fuel cells. Peroxides have a bleaching effect on organic substances and therefore are added to some detergents and hair colorants. Other large-scale applications include medicine and chemical industry, where peroxides are used in various synthesis reactions or occur as intermediate products. With an annual production of over 2 million tons, hydrogen peroxide is the most economically important peroxide. Many peroxides are unstable and hazardous substances. They cannot be stored and therefore are synthesized in situ and used immediately. In biochemistry, peroxides are usually very reactive and thus occur in nature only in a few forms. These include, in addition to hydrogen peroxide, a few vegetable products such as ascaridole and a peroxide derivative of prostaglandin. Hydrogen peroxide occurs in surface water, groundwater and in the atmosphere. It forms upon illumination or natural catalytic action by substances containing in water. Seawater contains 0.5 to 14 Argal of hydrogen peroxide. Freshwater 1 to 30 Argal and air 0.1 to 1 parts per billion. Hydrogen peroxide is formed in human and animal organisms as a short-lived product in biochemical processes and is toxic to cells. The toxicity is due to oxidation of proteins, membrane lipids and DNA by the peroxide ions. The class of biological enzymes called SOD is developed in nearly all living cells as an important antioxidant agent. They promote the disproportionation of superoxide into oxygen and hydrogen peroxide, which is then rapidly decomposed by the enzyme catalase to oxygen and water. Formation of hydrogen peroxide by superoxide dismutase, peroxisomes are organelles found in virtually all eukaryotic cells. They are involved in the catabolism of very long-chain fatty acids, branched-chain fatty acids, diamino acids, polyamines, and biosynthesis of plasmalogens, FR phospholipids critical for the normal function of mammalian brains and lungs. Upon oxidation, they produce hydrogen peroxide in the following process. FAD equals flavonidine dinucleotide, catalase, another peroxisomal enzyme, uses this H2O2 to oxidize other substrates, including phenols, formic acid, formaldehyde, and alcohol, by means of the peroxidation reaction, thus eliminating the poisonous hydrogen peroxide in the process. This reaction is important in liver and kidney cells, where the peroxisomes neutralize various toxic substances that enter the blood. Some of the ethanol humans drink is oxidized to acetaldehyde in this way. In addition, when excess H2O2 accumulates in the cell, catalase converts it to H2O through this reaction. Another origin of hydrogen peroxide is the degradation of adenosine monophosphate which yields hypoxanthine. Hypoxanthine is then oxidatively catabolized first to xamine and then to uric acid, and the reaction is catalyzed by the enzyme xamine oxidase. The degradation of guanosine monophosphate yields xamine as an intermediate product which is then converted in the same way to uric acid with the formation of hydrogen peroxide. Eggs of sea urchin, shortly after fertilization by a sperm, produce hydrogen peroxide. It is then quickly dissociated to O radicals. The radicals serve as initiator of radical polymerization, which surrounds the eggs with a protective layer of polymer. The bombardier beetle has a device which allows it to shoot corrosive and foul-smelling bubbles at its enemies. The beetle produces and stores hydroquinone and hydrogen peroxide, in two separate reservoirs in the rear tip of its abdomen. When threatened, the beetle contracts muscles that force the two reactants through valve tubes into a mixing chamber containing water and a mixture of catalytic enzymes. When combined, the reactants undergo a violent exothermic chemical reaction, raising the temperature to near the boiling point of water. The boiling, foul-smelling liquid partially becomes a gas and is expelled through an outlet valve with a loud popping sound. Hydrogen peroxide is a signaling molecule of plant defense against pathogens. In firefly, oxidation of luciferins, which is catalyzed by luciferases, yields a peroxy compound 1,2-dioxetane. The dioxetane is unstable and decays spontaneously to carbon dioxide and excited ketones, which release excess energy by emitting light. Hydrogen peroxide. The most widely used synthesis method of hydrogen peroxide is the anthraquinone process. There, anthraquinone is first hydrogenated to anthrohydroquinone. This reduced compound is oxidized with molecular oxygen, regenerating anthraquinone and releasing hydrogen peroxide. Direct synthesis of hydrogen peroxide from hydrogen and oxygen is inefficient and currently is not practiced industrially. Many peroxides of mineral acids, such as peroxidisulfates and percarbonates, can be obtained by anodic oxidation of the respective acids. The anode material must be stable to the required high potentials of a few volts and therefore is either platinum or its alloys. Peroxidisulfuric acid was historically used for the production of hydrogen peroxide in a method developed in the early 20th century. This process requires relatively high concentration of peroxidisulfuric acid as its more dilute solutions evolve oxygen gas instead of peroxide. Inorganic peroxides. Inorganic peroxides are divided into ionic and covalent peroxide. The first class mostly contains the peroxides of the alkali and alkaline earth metals, whereas the covalent peroxides are represented by such compounds as hydrogen peroxide and peroximonosulfuric acid. In contrast to the purely ionic character of alkali metal peroxides, peroxides of transition metals have a more covalent character. Defect formation in fluoride dioxides. Both CEO2 and THO2 accommodate excess oxygen in their lattice by forming peroxide molecules. This is in contrast to UO2, which takes in excess oxygen by forming charged interstitials. The accommodation of excess oxygen in these systems changes their thermal and diffusion properties. This will have implications in the oxide's use in nuclear fuels and solid oxide fuel cells. Adding oxygen to the CEO2 system, for example, will increase the diffusivity of the oxygen, allowing for more efficient solid oxide fuel cells. Peroxide salts, bonding in O22. The peroxide ion is composed of two oxygen atoms that are linked by a single bond. The molecular orbital diagram of the peroxide dianion predicts a doubly occupied antibonding I orbital and a bond order of 1. The bond length is 149 pm, which is larger than in the ground state of the oxygen molecule. This translates into the smaller force constant of the bond and the smaller frequency of the molecular vibration. The peroxide ion can be compared with other molecular oxygen ions superoxide O2A and ozonide O3A, but contrary to them, the peroxide is not a radical and not paramagnetic. Owing to the weak bonding between the oxygen atoms, peroxide easily undergoes homolytic cleavage, yielding two highly reactive radicals. This cleavage is accelerated by temperature, illumination or chemical reactions. Preparation of peroxide salts. Most alkali metal peroxides can be synthesized directly by oxygenation of the elements. Lithium peroxide is formed upon treating lithium hydroxide with hydrogen peroxide, 2Li plus H2O2 or Li2O2 plus 2 hours 2O. Historically, barium peroxide is prepared by oxygenation of barium oxide at elevated temperature and pressure. 2Ba plus O2 or 2Ba O2, barium peroxide was once used to produce pure oxygen from air. This process relies on the temperature-dependent chemical balance between barium oxide and peroxide, The reaction of barium oxide with air at 500 degrees Celsius results in barium peroxide, which upon heating to above 700 degrees Celsius in oxygen decomposes back to barium oxide releasing pure oxygen. Properties of peroxide salts. Few reactions are generally formulated for peroxide salt. In excess of dilute acids or water they release hydrogen peroxide. Upon heating, the reaction with water leads to the release of oxygen instead. The peroxide anion is a stronger nucleophile than hydroxide and displaces hydroxyl from oxyanions, for example forming perborates and percarbonates. Sodium perborate and sodium percarbonate are important consumer and industrial bleaching agents. They stabilize hydrogen peroxide and limit side reactions. The peroxide anion forms an adduct with urea, hydrogen peroxide, urea. Hydrogen peroxide is both an oxidizing agent and reducing agent. The oxidation of hydrogen peroxide by sodium hypochlorite yields singlet oxygen. The net reaction of a ferric ion with hydrogen peroxide is a ferrous ion and oxygen. This proceeds via single electron oxidation and hydroxyl radicals. This is used in some organic chemistry oxidations, for example in the Fenton's reagent. Only catalytic quantities of iron is needed since peroxide also oxidizes ferrous to ferric ion. The net reaction of hydrogen peroxide and permanganate or manganese dioxide is manganese ion. However, until the peroxide is spent, some manganese ions are reoxidized to make the reaction catalytic. This forms the basis for common monopropellant rockets. As a ligand in coordination chemistry, peroxide functions as a bidentate ligand in a variety of coordination complex. Some complexes have only peroxide ligands, for example, chromium, 6, oxide peroxide 42. Similarly, molybdate reacts in alkaline media with peroxide to form red peroxymolybdate Mo, O2, 42 euro. The reaction of hydrogen peroxide with aqueous titanium, 4, gives a brightly colored peroxy complex that is a useful test for titanium as well as hydrogen peroxide. Many transition metal dioxygen complexes are best described as adducts of peroxide. Applications. Many inorganic peroxides are used for bleaching textiles and paper and as a bleaching additive to detergents and cleaning products. The increasing environmental concerns resulted in the preference of peroxides over chlorine-based compounds and a sharp increase in the peroxide production. The past use of perborates as additives to detergents and cleaning products has been largely replaced by percarbonates in order to decrease the emission of boron to the environment. Sodium percarbonate is used in such products as OxiClean and Tide laundry detergent. When dissolved in water, it releases hydrogen peroxide and soda ash, 2Na2CO3 or 1.5H2O2 or 2Na2CO3 plus 3 hours 2O2. The use of peroxide compounds in detergents is often reflected in their trade names. For example, Persil is a combination of the words perborate and silicate. Some peroxide salts release oxygen upon reaction with carbon dioxide. This reaction is used in generation of oxygen from exhaled carbon dioxide on submarines and spaceships. Sodium or lithium peroxides are preferred in space applications because of their lower molar mass and therefore higher oxygen yield per unit weight. Alkali metal peroxides can be used for the synthesis of organic peroxides. One example is the conversion of benzoyl chloride with sodium peroxide to dibenzoyl peroxide. Organic peroxides. Organic peroxides can be divided into two major classes, peroxy acids and organic hydroperoxides. The first class is derived from the carboxylic acid and the second from ethers or alcohols. Preparation. Most peroxy acids can be obtained by the reaction of hydrogen peroxide with the corresponding carboxylic acid. R equals organic group, another route employs acyl halides instead of the carboxylic acid. This method is used primarily with aromatic compounds basic in order to neutralize the resulting hydrogen chloride. Aromatic aldehydes can be auto-oxidized into peroxycarboxylic acids. R equals aryl, the products, however, react with the initial aldehyde forming the carboxylic acid. Several synthesis routes are known for aliphatic peroxides, such as the reaction of dialkyl sulfates with alkaline hydrogen peroxide solution. In this method, the alkyl sulfate donates the alkyl group and the sulfate ion forms the leaving group. This method can also yield cyclic peroxides. The four-membered dioxetanes can be obtained by 2+2 cycle addition of oxygen to alkanes. He-selective synthesis of hydroperoxides can be carried out by free radical oxidation of alkanes with oxygen. Here the active site formed by a radical initiator reacts with oxygen to form a hydroperoxyl. The addition of oxygen results in a more active radical which can further extract hydrogen atoms and release the hydroperoxide, leaving a new radical. This process is used industrially for the synthesis of phenol from benzene and is called the Cumin process or Hock process for its cumin and cumin hydroperoxide intermediates. This auto-oxidation reaction can be used with common solvents from the group of ethers, such as diethyl ether, diisopropyl ether, tetrahydrofuran or 1,4-dioxane. It yields a volatile hydroperoxide ether that upon heating can result in a serious explosion. Peroxides are formed by living organisms through ene reactions or Diels-Alder reactions between alkanes and oxygen. Unsaturated fatty acids can serve as the olefinic substrates for the ene reaction and unsaturated amino acids like histidine can be the reactant for the Diels-Alder cyclization. Rancidification of fats is partly caused by the formation of peroxides. Properties and applications of organic peroxides. Pyric carboxylic acids are generally weaker acids than the parent carboxylic acids. Like most peroxides, they are strong oxidants and tend to explode at high concentrations and higher temperatures. Organic peracids are used in the synthesis of epoxies via the Prelise-Hiv reaction. Another important application is the synthesis of lactones of cycloctones in the Baeyer-Villager oxidation process. In both cases, electron-poor pyric carboxylic acids are especially efficient, such as metachloroperoxybenzoic acid. Tert-butyl hydroperoxide is a common oxidant in the Sharpless epoxidation, which is used for the stereoselective synthesis of epoxides. Carl Barry Sharpless was awarded the 2001 Nobel Prize in Chemistry for this reaction. Peracetic acid is a popular disinfectant in the medical field and food industry. Various peroxide solutions are commercially produced for the cleaning and disinfection of contact lenses. Dibenzoyl peroxide is used as a radical initiator. Its weak peroxide bond cleaves easily, yielding reactive benzoxy radicals, which assist polymerization leading to plastics like polyethylene. One of the synthesis methods of the commercially important plastic caprolactam the precursor to nylon 6 is a Baeyer-Villager rearrangement of cyclohexanone with peracetic acid. This yields caprolactone, which is then converted to caprolactam by reacting it with ammonia. Industrial resins based on acrylic and/or methacrylic acid esters are invariably produced by radical polymerization with organic peroxides at elevated temperatures. The polymerization rate is adjusted by suitable choice of temperature and type of peroxide. Some peroxides are drugs, whose action is based on the formation of radicals at desired locations in the organism. For example, artemisinin and its derivatives, such as artesunate, possess the most rapid action of all current drugs against falciparum malaria. Artesunate is also efficient in reducing egg production in Schistosoma hematobiome infection. Many organic peroxides can initiate explosive polymerization in materials with unsaturated chemical bonds, and specifically triacetone triperoxide and hexamethylene triperoxide diamine are powerful explosives. TATP is an inexpensive compound and is relatively easy to make. Whereas most other potent explosives, such as trinitrotoluene or RDX, contain nitrogen, which is relatively easy to trace by sniffing techniques, TATP is nitrogen-free and therefore is very difficult to detect by conventional screening methods. For this reason, it is an explosive favored by terrorists. TATP and HMTD were used in several executed or planned terrorist acts of the early 2000s, most notably in the 2001 shoe bomb plot and the 2005 London Underground bombings. Several detection devices have been designed since those events. One, for example, releases a chemical mixture which changes color when interacting with traces of TATP. Laboratory identification. Several analytical methods are used for qualitative and quantitative determination of peroxides. A simple qualitative detection of peroxides is carried out with the iodine-starch reaction. Here peroxides, hydroperoxides or peroxides oxidize the added potassium iodide into iodine, which reacts with starch producing a deep blue color. Commercial paper indicators using this reaction are available. This method is also suitable for quantitative evaluation, but it cannot distinguish between different types of peroxide compounds. Discoloration of various indigo dyes in presence of peroxides is used instead for this purpose. For example, the loss of blue color in leucomethylene blue is selective for hydrogen peroxide. Quantitative analysis of hydroperoxides is performed using potentiometric titration with lithium aluminium hydride. Another way to evaluate the content of peroxides in peroxides is the volumetric titration with alkoxides such as sodium ethoxide. Safety. Organic peroxides can accidentally initiate explosive polymerization in materials with unsaturated chemical bonds. Most notably, TATP and HMTD are high explosives, and TATP, because of its high susceptibility to accidental detonation by shock, friction, or sparks, has earned the nickname Mother of Satan among certain Islamic militant groups. TATP can accidentally form as byproducts in many commonly used reactions. These reactions range from synthesis of MDMA, where TATP is formed via isosafrole oxidation in acetone, to industrial production of phenol, where the second product of the Kuhlmann process, acetone, is partially oxidized to peroxide on the second reaction step. Accidental preparation of organic peroxides can occur by mixing ketone solvents with waste materials containing hydrogen peroxide or other oxidizers and leaving the mixture standing for several hours. In addition, many liquid ethers in the presence of air, light and metals slowly over a period of months form highly unstable ether peroxides such as diethyl ether peroxide. Therefore, ethers are often stored over potassium hydroxide, which not only destroys peroxides but also acts as a powerful desiccant. Peroxides are also strong oxidizers and easily react with skin, cotton and wood pulp. For safety reasons, peroxidic compounds are stored in a cool, opaque container, as heating and illumination accelerate their chemical reactions. Small amounts of peroxides, which emerge from storage or reaction vessels, are neutralized using reducing agents such as iron, sulfate. The safety measures in industrial plants producing large amounts of peroxides include the following. The equipment is located within reinforced concrete structures with foil windows, which would relieve pressure and not shatter in case of explosion. The products are bottled in small containers and are moved to a cold place promptly after the synthesis. The containers are made of non-reactive materials such as stainless steel, some aluminium alloys or dark glass. History. One of the first synthetic peroxides, barium peroxide, was synthesized by Alexander von Humboldt in 1799 as a byproduct of his attempts to decompose air. 19 years later, Louis-Jacques Tharp copyright now recognized that this compound could be used for the preparation of a previously unknown compound, which he described as oxidized water, now known as hydrogen peroxide. Sodium peroxide was synthesized in 1811 by Tharp, copyright Nard and Joseph Louis Gay-Lussac. The bleaching effect of peroxides and their salts on natural dyes became known around that time, but early attempts of industrial production of peroxides failed, and the first plant producing hydrogen peroxide was built only in 1873 in Berlin. The discovery of the synthesis of hydrogen peroxide by electrolysis with sulfuric acid had brought the more efficient electrochemical method. It was first implemented into industry in 1908 in Wittgenstein, Carinthia, Austria. The anthraquinone process, which is still used, was developed during the 1930s by the German chemical manufacturer IG Farben in Ludwigshafen. The increased demand and improvements in the synthesis methods resulted in the rise of the annual production of hydrogen peroxide from 35,000 tons in 1950, to over 100,000 tons in 1960, to 300,000 tons by 1970, and by 1998, it reached 2.7 million tons. See also. References